Deoxyribonucleic acid is the molecule used to store the genetic information of any organism. It's found in the nucleus of eukaryotes and cytoplasm of prokaryotes as highly coiled protein-containing structures called chromosomes. A unicellular organism usually has one circular chromosome that is secluded to an area of the cytoplasm, as well as a few smaller circular DNA molecules. Multicellular organisms have more than one chromosome. These are linear and secluded to the nucleus of the cell. Most non-cellular organisms, such as viruses and bacteriophages, also possess a genome composed of DNA. But some viruses, such as Retroviridae, Flaviviridae, and Rioviridae, exist as double-stranded RNA or single-stranded DNA genomes. DNA is a long chain assembled from 4 nucleotides called adenosine monophosphate, guanosine monophosphate, cytidine monophosphate, and thymidine monophosphate. In RNA, thymidine monophosphate is substituted for by uridine monophosphate. The 3 main components of a nucleotide are a nitrogen-containing base, an oxidized pentose sugar, and a phosphate group. The bases of the nucleotides are called adenine, guanine, cytosine, and thymine, and abbreviated to A, G, C, and T when written as a sequence or primary structure. In RNA, thymine is substituted for by uracil, which is abbreviated to U. A nucleoside is a nucleotide that lacks the phosphate group. The bases of the nucleosides are called adenosine, guanosine, cytidine, thymidine, and uridine. Each base is covalently bonded to a sugar that is linked to a phosphate group. The sugar-phosphate compound is hydrophilic and remains in contact with the aqueous environment. The bases are hydrophobic and will form favorable interactions with other hydrophobic molecules, such as the bases of another strand. The sequence of nucleotides defines the amino acid sequence of the proteins, regulates transcription and replication, DNA-protein interactions, defines the structure of the molecule, and how it is arranged within the nucleus or cytoplasm of a cell. DNA. The sugar of one nucleotide and the phosphate group of another undergo an esterification reaction linking one nucleotide to the next to form a polymer or strand of DNA. The 3' hydroxyl group on the sugar of the first nucleotide in sequence forms an ester link with the 5' phosphate of the second nucleotide in sequence, whose 3' hydroxyl in return has a bonding reaction with the phosphate group on the next nucleotide. The phosphate group attached to the 5' carbon of the deoxyribose of the first nucleotide in sequence is exposed, and this nucleotide is referred to as the 5' start of the molecule. The hydroxyl group on the 3' carbon of the last nucleotide is also exposed, and referred to as the 3' end of the DNA molecule. The sequence of nucleotides is assembled in the 5' to 3' direction so that the first nucleotide in the polymer always has an exposed phosphate group and the last nucleotide an exposed hydroxyl group. A strand of DNA will form a double-helix with another DNA strand whose sequence is in the antiparallel 3' to 5' direction so that the 5' start of one molecule is the 3' end of the second molecule. The lagging strand, which is antiparallel to the leading strand, has to be complementary in sequence in order for the double helix to form. DNA replication and the transcription of genes occurs in the 5' to 3' direction. Each strand of the helix is assembled according to the mechanism described previously. The sequence replicated from the leading or template strand is in the 3' to 5' direction and will form the antiparallel strand of the new chromosome once replicated. Because the antiparallel or lagging strand is in the 3' to 5' direction and cannot be replicated in the 5' to 3' direction, it is replicated in shorter sequences referred to as Okazaki fragments, which are then ligated as they are replicated into a longer polymer that forms the leading strand of the new chromosome. As a result, the two strands produced by DNA replication are identical to their template strands. During transcription of shorter sequences, such as that of genes, the strand produced is also assembled in the same manner as it occurs during DNA replication, except that thymidine monophosphate is substituted for by uridine monophosphate. In the 1950s, James Watson and Francis Crick from the University of Cambridge produced an accurate model of the secondary structure of DNA and discovered the mechanism of base pairing. Watson and Crick created an accurate structural model of the DNA double helix using data published by a number of scientists, including Rosalind Franklin from King's College London and Edwin Chargaff from Columbia University. Chargaff had found out that the bases adenine and thymine and the bases cytosine and guanine occur in a 1-to-1 ratio, and Franklin was conducting structural studies of DNA using X-ray crystallography at the time, where a crystallized and suspended DNA molecule is subjected to X-rays that map the location of the atoms. Watson and Crick then constructed an accurate three-dimensional model of the DNA double helix and demonstrated that this structure is stabilized by the fact that adenine forms 2 hydrogen bonds with thymine and guanine forms 3 hydrogen bonds with cytosine. They coined it the Watson-Crick base pairing mechanism and received the Nobel Prize for their contributions. The base of any nucleotide will dissociate and gain a proton when the pH is below its dissociation constant, the pKa, and lose a proton at a pH above that value. The purines adenine and guanine are constructed from 2 conjoined cyclic rings, of which one is a pentose and the other a hexose. The 2' nitrogen of adenine has a pKa of 3.88 and will gain a proton at a pH below that value and lose a proton at concentrations above that value. Guanine has 2 atoms that can dissociate to gain or lose a proton. The 2' nitrogen dissociates at a pH of 10, and the 7' nitrogen on the pentose dissociates at a value of 3.60. The 3 pyrimidines, cytosine, thymine, and uracil, are all hexoses and all have a 2' nitrogen that can gain or lose a proton. Cytosine has a pKa of 4.56, thymine has a pKa of 10.50, and uracil a pKa of 10.10. The ability of the bases to dissociate allows RNA to participate in chemical reactions with other molecules and for complementary bases on opposite strands to participate in hydrophobic base pairing interactions. Unfavorable energy-requiring reactions can displace the protons of bases to different positions and result in the formation of their tautomers. In adenine and cytosine, such a reaction will displace a proton from the amino group attached to the first prime carbon to the second prime nitrogen of the base, changing it from the regular amino to the imino tautomer. The keto form of the bases guanine and thymine convert to the enol tautomer when a proton from the second prime nitrogen is displaced to the oxygen atom of the first prime carbon and forms a hydroxyl group. Only less than 0.01% of nucleotides will be in their tautomeric form because reactions that require cofactors such as NAD are unfavorable. Proton displacement changes the conformation that the bases adopt and leads to errors during DNA replication, which consequently contributes to genetic variation. The difference between deoxyribonucleic acid and ribonucleic acid is the conformation of their sugars.
In RNA, both the 2' and 3' carbon of the pentose are reduced, and then the 2' endo conformation, where the 2' carbon is elevated and the 3' carbon ascends. The pentose in DNA is in the 3' endo conformation, where the 2' carbon is oxidized and ascends, whereas the 3' carbon remains reduced and elevated. Nucleotides can only assemble into either a DNA or an RNA polymer because if both types of nucleotides were present in the same polymer, their sugars would collide. However, complementary bases on a DNA and RNA polymer can still interact and form a double-stranded helix. This makes it possible for the cell to carry out processes such as transcription or reverse transcription in the case of RNA-based viruses.
DNA helices can adopt 3 types of secondary structures called the B helix, the A helix, and the Z helix. The most common of these is the B helix. All of the helices have a diameter of 20 angstroms. One helical turn has 10.5 base pairs and has a length of 36 angstroms. The minor groove in B DNA has a width of 9 angstroms, whereas the shallow major groove is 13 angstroms wide and is lined with atoms and chemical groups that participate in reactions. The A helix is mostly induced by DNA binding proteins and is the only structure that double-stranded RNA or an RNA-DNA complex can adopt. The minor groove in the A helix is greater than 9 angstroms, making it wider and shallower than in the B helix, whereas the major groove decreases to become narrower and deeper, preventing previously exposed atoms and chemical groups from participating in any chemical reactions. An A-type DNA helix is not in a complex with a protein, will supercoil in order to return to a favorable state of 10.5 base pairs per helical turn. The Z-type helix is left-handed and a very uncommon structure that DNA can adopt in vitro as a result of regions rich in GC repeats, cytosine modifications, methylations, and a high salt concentration. The tertiary structure of DNA is its supercoiled form. Supercoiling increases the capacity of the cell to store information because it compresses long linear DNA into compact and coiled chromosomes that fit easily into the nucleus or cytoplasm of a cell. Supercoiling is carried out by enzymes called topoisomerases, which cleave and then ligate DNA once another enzyme, such as a helicase, has either twisted or unwound the molecule. In mammals, topoisomerase II cuts both DNA strands, and topoisomerase I cuts only one strand. The bacterial ortholog is called topoisomerase IV and cuts both strands like topoisomerase II in mammals. The degree of chromosomal supercoiling can be determined by looking at the relationship between the linking number, twist, and writhe. The linking number is the sum of the twist and writhe and refers to the number of times a DNA strand makes a full right-handed turn around the axis of the helix. A full right-handed turn around the helix is made every 10.5 base pairs, so a molecule of 1,260 base pairs has a linking number of 120. The twist refers to the winding of the DNA strands around each other. For every 10.5 base pairs, there is a twist of 1, so that a molecule of 378 base pairs has a twist of 36. The writhe is the measure of the supercoiling of the axis of the double-stranded helix. A positive writhe indicates that the supercoils are left-handed, a negative value that the supercoils are right-handed, and a writhe of 0 that there are no supercoils and that the molecule is relaxed. Topoisomerases make it possible to separate tangled chromosomes and DNA during replication, transcription of genes, and other processes. When constrained linear DNA is supercoiled, the linking number initially changes according to the number of turns that have been added or subtracted. It then remains at that value throughout the process of supercoiling, while both the twist and writhe will increase or decrease relative to it. For example, a constrained and linear DNA strand with a linking number of 30, twist of 30, and no supercoils is cleaved by a topoisomerase and unwound by one helical turn. Both the linking number and twist decrease to 29 upon unwinding. Upon reannealing the strands, the torsional strain in the DNA molecule causes it to twist on itself, changing the twist to 30 and adding one negative supercoil or writhe of -1. Twisting on itself returns the molecule back to a favorable state of 10.5 base pairs per turn. The linking number does not change and is the sum of the twist and the writhe. Supercoiling with closed circular DNA molecules works the same as with constrained and linear DNA molecules. Upon being cleaved by a topoisomerase, the molecule becomes linear and unconstrained. In this example, the molecule is then unwound by two helical turns, and the linking number and twist both decrease from 30 to 28. Upon ligating the ends to form closed circular DNA again, the tension in the molecule causes it to twist on itself and form two negative supercoils. The twist returns to 30, the writhe becomes -2, and the molecule returns to a favorable state of 10.5 base pairs per turn. The twist is increased by the number of turns it's unwound by, and the writhe decreases by the same number, so that the linking number is the sum of the twist and the writhe. Supercoiling allows for greater amount of information to be stored within a cell and also protects DNA from damage when exposed to deteriorating conditions.
Coenzymes are nucleotides with different side chains and chemical groups that can participate in a range of biochemical reactions. They store energy derived from metabolism, provide the energy, and regulate chemical processes. Adenosine triphosphate, or ATP, phosphorylates enzymes to activate or deactivate them and supplies unfavorable reactions with energy. It is produced during the breakdown of sugars and is involved in both the TCA cycle and electron transport chain. NAD and NADP act as oxidizing agents in the TCA cycle. Apart from being involved in the production of ATP, NAD also functions as a cofactor of the enzyme lactate dehydrogenase during the conversion of lactic acid to pyruvic acid. NADP is the phosphorylated analog of NAD that is mainly involved in carrying out the reverse reaction as a reducing agent that donates protons. Both coenzymes participate in cellular respiration. The coenzyme acetyl-CoA activates and transfers acyl groups and is involved in the breakdown and production of fatty acids. The coenzyme S-adenosylmethionine is involved in methylation of molecules such as DNA bases and can repress the transcription of genes, replication of sequences, or is used to mark and differentiate a genome from that of another if present in the same cell, such as during viral infection. Like NAD and NADP, the cofactor FAD can also participate in the reduction and oxidation reactions. Purified cofactors are often sold together with enzymes for use in the academic or industrial research laboratory. DNA stores genetic information as a long chain of 4 nucleotides: adenosine monophosphate, guanosine monophosphate, thymidine monophosphate, and cytosine monophosphate. The sequence of these bases forms the primary structure of DNA. A nucleotide consists of a nitrogen-containing base, a deoxyribose, and a phosphate group. The pentose sugar of DNA has a 3' endo conformation, whereas the pentose of RNA has a 2' endo conformation. Both sugars cannot be present in the same strand because their conformation would cause them to collide and disrupt the helical structure of DNA. The secondary structure of DNA was discovered in the 1950s by James Watson and Francis Crick. They figured out the hydrophobic base pairing mechanism that joins complementary bases of different strands. The Watson-Crick base pairing mechanism stabilizes the double helical structure of DNA. The bases of nucleotides possess protons that dissociate at pH values above or below their pKa and may form tautomers. DNA adopts three types of secondary structures: the B helix, A helix, and Z helix. The B helix is the most common structure adopted by DNA. The A helix is the only conformation DNA, RNA, or RNA molecules can adopt, and the Z helix is only adopted in vitro under certain conditions. The tertiary structure of DNA is facilitated by supercoiling. This process forms highly condensed structures which reduce the space required and increase the amount of genetic information stored. Supercoiling can be negative where the molecule is twisted on itself, or positive where the molecule is unwound to create or release torsional strain. Coenzymes are nucleotides that have additional phosphate groups, such as an ATP, or possess other chemical groups that are involved in carrying out chemical reactions. DNA is a molecule whose properties make it very effective at storing and permitting access to genetic information.